Welcome to today's class of plant developmental biology, where we are going to study how to study plant developmental biology. So, one of the very important and prominent approach which we take to study plant developmental biology is molecular genetics based approach. So, molecular genetics is basically to study how a particular gene in a genome regulates a particular developmental process in the plants. So, there are two major approaches in the molecular genetics which we usually take to study plant developmental biology. One is the forward genetics based approaches and the reverse genetics based approaches. So, in forward genetics what we do, we begin with a mutant or with a phenotype and then try to identify what is a gene associated with that phenotype. Whereas in case of reverse genetics, first we define our gene of interest and then we try to understand or we design experiment to pinpoint what is a specific developmental function of that gene. So, you can look this schematic diagram. So, in forward genetics we start with the phenotype and then we map the associated gene or the genetic loci which is associated with this phenotype. Whereas in case of reverse genetics, we have gene, we identify the gene first and then we try to understand, does this gene has any role to play in the plant development, and if it is so, what is its specific function in the plant growth and development. So, now we will take one by one. So, first we will discuss more about the forward genetics based approaches. So, as I said, the first step of the forward genetics is that you should have a mutant. Mutant is very important. So, or any variant. So, mutant means a particular defect in the development. So, defect is important because if you do not have abnormality, it is difficult to trace what is the normality or what is the mechanism which is regulating the normal development. So, typically what are different steps which we follow during forward genetics based approach. The first approach is identifying a desired developmental phenotype or a mutant with a phenotype. Then once we identify this mutant, then obviously we are going to analyze genetics or inheritance pattern of this mutant, whether it is dominant, recessive, semi-dominant. And then once we know the pattern of the mutant or the inheritance pattern of the mutant, then we go and we try to map the genetic loci. So, this is important, then try to understand that what is the genetic loci which is actually responsible for this developmental defect. And then final step in forward genetics what we take, we then validate the genotype and phenotype association. So, how we do? The first step, normally we take a mutant or any variants, natural variants are also a way to study, and then we finally do the complementation to validate this one. So, the first important thing in any study of plant developmental biology is to fix your biological question. So, if you recall our previous classes, so what we have discussed, we have referred what are the different developmental events which takes place during the process of plant growth and development. So, depending on that you can first choose your question. So, you can define that which aspect of development you want to study. For an example, are you interested in studying what are the genetic loci which is responsible for maintaining the meristem? Second, developmental events what you can study is the organ identity and patterning. So, you want to understand that what are the genes or what are the genetic element which is responsible to define a particular identity of a organ, of a tissue. And the second thing is that you can identify what is the genes or what are the genetic elements which is responsible for patterning. This is important in the process of plant architecture maintenance. Then you can identify mutants which shows abnormality in shape or size of a particular organ, particular part of the plant. And then another very interesting thing what you can study, what are the developmental mutants which affects phase transition. So if you recall again our previous class, so we have already discussed that during growth and development a plant passes through different phases, starting from the embryogenesis phase. Then in the post embryogenic, first it undergo the process of juvenile development, then there is a transition from juvenile to the adult vegetative development, and then from adult vegetative development there is another transition or major transition is from vegetative to reproductive development. So these phases are regulated by in the time. So what we can do, we can identify a variants or a mutants where these stages or these transitions are defective. Another important thing, if you recall previous class, we have talked about the activation of branching or activation of secondary growth. So these both these processes are regulated and it occurs only at a certain time point. So it is also important, as you can imagine that they are also regulated by genetic element. So we can also identify or screen a mutant where these processes are defective. So now we'll go and look what are the different ways to identify the mutant. So the first step of forward genetics we are taking here to identify a desired developmental phenotype or desired mutant. The first and maybe more important with respect to the breeding approaches was to look the natural variation. So in this nature there are a lot of variations in the developmental stages, organs, structure, function, everything. So if you look for example this upper panel, so here are different accessions of Arabidopsis thaliana, and what you look, there are variation, morphological variation. So if you look their leaves, the number of leaves, shape of leaves, size of leaves, so there are huge amount of variation even within a same species. So what we can do, we can choose any one of these two contrasting two accession with the current contrasting or with the different trait, and then you can try to identify the associated loci. If you look here, so this is basically variations at the 6th rosette leaves, so across different accession. So you can see that even not only the overall morphology but shape and size of a specific organ also shows a large amount of variation across the species or across the accessions. Here is the variation in the architecture. So you can look, these all plants are compared at the time of after transition, floral transition has occurred, and you can see that across these different variants or different accessions there is a large amount of variation in their overall or final root final plant architecture. So this could be one source where you can identify a particular trait of interest and you want to study the associated gene. Second way of moving is you can induce the mutation. So you can basically create a mutant plants where you can look for a special phenotype. So there are many ways to do that. We are not going to talk more about it, but what we can do, we can take few example of very widely used way to study the to generate the developmental mutants. So there are three major category: chemical mutagens, physical mutagens and biological mutagens. In chemical mutagens, the most frequently used chemical is EMS. EMS, it generates a point mutation. So what happens, you will see in the next slide that it creates a kind of change the nucleotide in a way that GC base pair getting converted into AT base pair. So this is a kind of point mutation, and this any point mutation has a lot of benefit. Second mutagen is physical mutagens, where you can use different electromagnetic radiations, gamma rays, X-rays, UV light, and they can induce random mutations in the genome. And we will take an example of a gamma rays radiation. Third way of generating mutants are biological mutagens, where we are using a bacteria which is called agrobacterium, and agrobacterium basically we are using this agrobacterium mediated transformation system, where we can use either tDNA or transposon based system, and we want to create a kind of gene knockout by insertional inactivation. So again, if you look how EMS works, so as I said, so let's assume here is a GC base pairing, and if you treat with the EMS, what it does, it changes, it modifies this nucleotide, the G, and it adds methylene. And once this modification has occurred, in the next round of replication, this methylated G cannot pair with the C, instead it can pair with the T. So now it has added T in place of C in one strand, and once this DNA will undergo the second round of replication, at the place of T, naturally A will be added. So if you look from here to here, there is a transition of CG base pair to TA base pair. This is creating a kind of point mutation, and this is one of the very powerful chemical mutagen which we are using, or people who are working in the field of developmental biology is extensively using to generate point mutations. So what you do with this one? So the way of generating, let's take what typical example of Arabidopsis thaliana. So you take a large number of seeds and expose these seeds with the chemical, and what you are expecting a random mutations in some of the seeds anywhere in the genome. You cannot control it, and then you generate a population where you will have some non-mutants, some mutants, and whenever this mutation will occur in the germ line, and you take these plants, grow them, and when you grow them, there is a possibility, if your plant has come from the non-mutated seeds, it will have a normal, it will create a normal pattern, but there is a possibility that you screen a plant which is coming where the genome has been already mutated. So you have generated some kind of point mutations, and as I said that this way of generating mutation is totally random. So you have to screen a very large population of mutant to find your desired mutant. Then you screen this population and look for your phenotype, fix your phenotype. So first thing, let's assume that if you want to study the root development, then you look a mutant where root is defective. If you want to study a shoot development, find or screen for a mutant where shoot is defective, flower is defective, whatever your interest is, you can fix it and then try to identify a mutant phenotype. And we know from the genetics, this mutant can be either dominant, recessive, it can be either in homozygous condition or in heterozygous condition. These all kind of things you can analyze, and you can and understand that what is the pattern. So I will take some examples. So if you look here, this is Arabidopsis root. This is a typical or very early stage Arabidopsis seedling. When this seedling was EMS mutagenized, and it was and people were looking for some kind of defects in the root, and what we observe that if you look this, the shoot developments look quite normal, but there were 3 independent mutants which was identified from this population where root development was affected. So you can see they are very short root. Not only that, this is very powerful, you can even make a very sophisticated genetic screen. For example, if you look these 2 pictures, so here we want to identify the tissue patterning in a very special tissue which is called phloem. So if you look this signal, this is a protein, green fluorescent protein, and what happens that if you express or if you produce this green fluorescent protein in the phloem cells, it can move through the phloem cells and it get distributed in the root tip. Now if someone want to identify what are the genetic locus or what are the gene which is responsible for giving a proper identity to the phloem cell or regulating a proper differentiation to the phloem cell, you can mutate these plants and look the plant where this property of phloem is affected. So if you look the mutant, here you can see that we have the green fluorescent signal somewhere here, but somehow the fluorescent is not moving, the GFP is not moving. So this tells that in this mutant some aspect of either phloem developments are affected or phloem function which is transporting the molecules are affected. So you can fix your biological question here, and then you can go and trace back what is the genetic loci associated with this one. Okay, similarly, this mutagenic approach you can tell, let's assume that you already have a mutant, you know function of a particular gene through the forward genetics, you can take further and you can try to identify other genetic component of that entire pathway by mutagenizing your mutants and screening for second site mutations, identifying enhancer, identifying suppressor. So, you can take these examples here. So, this is a gene which is called AG, agamous gene. So, in this mutant, this is a single mutant, you can see some phenotype. The phenotype is indeterminate floral meristem, no carpel, but stamen is fine. When this mutant was mutagenized and look for another second site mutation, so there was another mutant which was identified, HUA1, HUA2, and these mutants along with the agamous 4 mutants, double mutants. If you look, all other the mutant phenotype is still there, but even stamens are getting converted into petal. So, either second site mutation can enhance the first mutation. Similar case you can hear HUA1, HUA2 alone, they do not have any phenotype. So, if you take a single mutant, there is no obvious phenotype, but when you screen for another higher order mutants, so HUA1 and 2 with HEN1, there is a loss of floral determinacy, there is a homeotic conversion of stamen into petal. Similarly, HUA1 and HUA2 with AGO10 gene, there is a loss of floral determinacy. With agamous, there is loss of floral determinacy and conversion. So, if you look this is a way, so this provides a very powerful way to perform a very special or very targeted genetic screen and identify double mutant, triple mutant, and then try to understand the entire genetic pathway. You can trace out the genetic pathway, what all genes are responsible to regulate a particular process of development. Another important thing, if you look here, this is a screening a suppressor. So, let us assume that I have identified a mutant where leaf is defective. If you look here, this is wild type, there are so many leaves and shape and is different, but when you have a mutant where number of leaves and the shape and size of leaves are altered, now I have identified one gene or one genetic component which is responsible for this patterning. I want to identify what are the other gene or what are the other genetic components which are interacting with this gene to provide this particular shape and size. You can take this mutant and now you can look for double mutant where even the previous mutants are getting suppressed. This is called suppressor mutants. In these cases, you are adding them and you are identifying more severe mutant phenotype. Here we are looking for a mutant where even the existing phenotype is disappearing or getting suppressed. So, this technique you can use to identify both kind of genes. Okay, I will not go in detail, but you can look this picture here, gamma mutagenesis, physical radiations being used to mutagenize the plants, and what you can see that there are lot different kinds of mutants with different phenotype. You can see chlorophyll phenotype, leaf phenotype, shoot phenotype, flower phenotype, all these kind of phenotype or mutants can be generated with this mutagenesis. So, these are the two way of doing mutagenesis, and third, another very important way to generate a desired developmental mutants is biological mutagen. Here there are three major approaches which is being taken place. First one is tDNA mutagenesis, which is done by agrobacterium mediated tDNA insertion. Second is transposon based mutagenesis, and third is activation tagging based mutagenesis. So, in transposons, we all you have studied in your course or previous course that transposons are the mobile genetic elements which naturally present in the organism, and they can actually jump in the genome. That is why they are also called jumping genes. So, we can use them as a technique to basically disrupt a particular gene and identify the mutant phenotype. So, what we can expect through this one? So, here basically this process is called insertional inactivation or insertional mutagenesis. So, what happens? So, either you take transposon or you take activation tagging. Activation tagging is slightly different, but if you take tDNA or transposon, so what basically you are doing, you are allowing this molecule to randomly get inserted in the genome, and if it happens that it got inserted in the gene or the regulatory elements of the gene, it can affect the expression or function of the gene, and that results in some mutant phenotype. So, these are some possibility. Let us assume that if this got inserted in the coding region, it may disrupt the coding region and you might get a loss of function phenotype. So, where the gene is disrupted, no functional proteins are made, and that is why you will have a phenotype because of the loss of the genes. Second possibility is that if the insertion somehow happens in the promoter region or untranslated region. So, promoter regions, you know, this is very important to regulate expression and expression pattern of a gene. UTR region, untranslated region, they directly are not involved in coding the protein, but lot of regulations occurs through the untranslated region. So, if there is insertion or disruption because of the tDNA or transposon in this region, what we expect? We can expect either reduced expression or altered expression pattern, and in both the case we might have the phenotype. Another way is that it happens that somehow the insertion happens in a gene or in the genetic elements, and it can also enhance the expression. This is more relevant when we talk about the activation tagging, but it has been also seen that if insertion happens somewhere in the promoter or it happens in an element which is like a silencer or repressor of a gene, and if this insertion suppress or kills the regulators, then you might have increased expression. But again, as I said that these processes are totally random. So, there is also possibility that there is a multiple insertion in the same genome at the multiple site, and this may generate a multiple knockout or knockdown, whatever is the condition. Okay, so we will briefly discuss how tDNA mutagenesis occurs. So, Agrobacterium is a bacteria which basically is known to infect the plant cells, and it has a plasmid which is called Ti plasmid. Plasmid, you know, it is extra chromosomal genetic material, and what happens that when the bacteria infect a plant cell, a section, a portion of this plasmid, Ti plasmid, which is called tDNA, is transferred in the plant cell, and then this tDNA get integrated in the plant genome, but that integration is totally random. So, it can go and integrate anywhere. So, if you want to use this technology and if you want to create, and there are certain elements which is maybe more relevant in the making transgenic, we might discuss in future, and but what happens here that if you can genetically engineer this tDNA, you can basically use this technology to generate mutants. So, what you do in this left border and right border, you can put either a visible marker, this will help you to identify the plants where insertion has occurred, or you can put a kind of selection marker. Selection marker, you can put an antibiotic resistance gene. So, you can basically directly select the transformant using this antibiotic, and then use this and generate a large number of transgenic plants, and then screen them either based on the visual selection marker or by selecting on the antibiotic. And this has been very extensively used in model plant Arabidopsis thaliana, and in fact, a full fledged collections of this tDNA insertion plants or mutants has been generated, and it has been stored or maintained as a form of a big program where anyone across the globe can order these lines, they can use for their own specific study. So, if you look how they look, so typically this is how it happens. So, if you take one example, this is one gene, and what it has been found that through the process of tDNA insertion, multiple site insertion has been generated. So, in some mutants it was inserted here, in some mutant it was inserted here, 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 here. So, you can say that for this gene at least 6 allele has been generated through the tDNA insertion, and all the 6 mutants has phenotype. So, if you look, this is a normal wild type plant, and the mutants, this is 1, 2, 3, 4, 5, 6, depending on their site of insertion there is a variations in the phenotype. So, all these mutants, they can be simply generated by integrating the tDNA. So, second way of generating mutation is transposon based mutagenesis. One very commonly used transposon is ACDS based. So, what happens, DS is the element of the transposon which actually jumps, which get excised from one place and get integrated to the another place, and AC is the part which helps in the process. So, what you can do, you can generate a TI plasmid, you can engineer a TI plasmid, and you can put AC here in one TI plasmid in agrobacterium, you can put DS in another TI plasmid in the agrobacterium, and what you do, you can generate independent transgenic lines, one for AC elements, one for DS element. The important thing is that DS element cannot jump until and unless AC element is present. But the good thing is that AC element can work in trans, so they need not to be together. Even though if they are present in the same plant at different genetic loci, it is fine, they can still work. And then what you do, you take this plant, and if you cross them, you are basically bringing both AC element as well as DS element in the same transgenic plant, and once this AC is available, this DS will start transposition, and during this transposition it can go and it can inactivate or it get inserted or integrated at different genetic loci, and which might create a developmental mutant. See the example here. So, using this example, this is maize plants, some mutants had been generated. If you look the wild type plants, these mutants, the leaves are not looking normal, this is more kind of drooping leaf, they are falling off. Okay, another very important transposon which is being used for generating mutation is TOS17. TOS17 is a retrotransposon, and it is very important because this transposon normally is stable at the adult plant or at the later stage of development. So, once it is, the element is present, but it is not very actively undergoing the process of transposition. But if you generate a callus from these plants, at callus stage these transposons get activated and it is started jumping. So, particularly for those plants where we can generate a transgenic plant using callus, it is very good way to generate a lot of insertional mutation for this one. Rice is one very good example of it. So, what you can do, you can make a callus, in the callus the transposition will occur, different genes or different loci will be disrupted, and then use this callus to just simply regenerate through the process of tissue culture and make transgenic plant. In this transgenic plant, you can identify a developmental defect, developmental mutant, and you can generate a collection of such kind of mutants. Here is few examples in rice which using this TOS70 retrotransposon, lot of mutants had been generated. This I am showing only few of them. You can look here, this is thick culm, then you have a kind of abnormal flower, you have endless stellar mutants, you have a very dense panicle. Panicle is basically inflorescence. Dense panicle means you have more panicle branches, more number of flowers, more number of seeds. So, these all mutants, they have been generated by using TOS70 based retrotransposition. Both of these approaches, normally the ACDS based approaches or tDNA based approaches, normally or usually they generate loss of function mutants because they might kill the genes. They are more prone to inactivate a gene. But another way to study a gene function, that if you can enhance the expression of a particular gene, and how can you enhance? So, here this is called gain of function. One way of enhancing is that you do not change their domain of expression. So, they still express in the same tissue where they normally express, but their expression level goes up. Second way is that if you change their domain of expression, or you can make them to express in a cells or in the tissue where they were not expressed earlier. And if this kind of genetic regulators are sufficient to give a proper identity or proper developmental events, if they are sufficient to initiate that kind of program, they will give a different phenotype. And this is being used through a process which is called activation tagging. So, what happens in the activation tagging? Random insertion of multiple enhancers. So, enhancers are the genetic elements which can increase the expression of a gene. So, what people are doing? They are using multiple copy of enhancer and they are genetically engineering a tDNA or a TI plasmid, and they are using this enhancer through agrobacterium mediated transformation, and they are generating a large number of plants. And then there those plants, they are looking if there is a plant where there is a phenotype, and most likely this phenotype could be because of the activation of a gene. So, if this enhancer elements, let us assume it gets inserted somewhere here very close to promoter of a particular gene or the regulatory elements of this particular gene, there is a possibility that because of its enhancer activity, it will enhance the expression of this gene or this gene, and you might have a developmental phenotype in this case. This is one example where activation tagging has been used to generate a mutations. As you can see, this is the site of mutations, and these mutations can actually increase the expression. So, if you look, this is a wild type, this is RT-PCR. We will discuss sometime in other class how we do exactly, but this is to measure, to quantify the messenger RNA level of a particular gene. So, if you look this gene, and if you take the expression level in the wild type, these genes are expressed at a low level, but when there is a activation tagging or insertion of the enhancer element in between, you can see the expression level of these genes are significantly increased in these lines. Okay, so we will stop here this class. In the next class, we will take this further and we will discuss if we have a mutant, how can we go further and try to identify the genetic loci associated with it. Thank you very much.